glad you're back for our discussion of what can change relative fitness and therefore the strength of selection. In your simulation that you worked through earlier, we were playing with things like relative fitness and starting allele frequency and then thinking about drift. And we're going to come back and fold drift back into this conversation in more detail in a bit. But for right now, I'm going to focus on the question of what can influence the relative fitness of an allele and therefore influence the selection, right? So if there's a big difference in relative fitness, then you're going to have stronger selection. So the way that I've been kind of explaining this, and we've gone through this in our meetings this week, but just to kind of simplify, I can remember how to do this. There we go. So let's have our imaginary scenario, however you want to say that, where we have an environment, a substrate, rocks or grass or whatever, moss, that's blue. And then we have organisms that live on that, and they're all red. So everybody's red. Everybody who lives, everybody in this whole population, every single one of them. And through random mutation, not because something pressured the mutation to happen. This, again, remember, mutation just happens. It just occurs. And through random mutation over the course of generations, an allele appears, a new allele. And that allele causes individuals to be blue. So I'm drawing a blue dot down here in the bottom. I don't know if you can see it because that individual is so well camouflaged, right? You can't see it. So if we're just doing a simple predation model as our example here. So now, predators can see the red individuals really well, but really can't see the blue individuals at all. So if, in our imaginary scenario, the red individuals are homozygous for the red allele, because that was the only one that existed, right? They could only have the red allele. And then let's imagine that our new mutation gives this individual a blue allele, so a B. I don't know if you can see that. So this individual, instead of being RR, is RB. Okay. So now, that individual is blue, everybody else is red. So if we just think about in this generation, what's going to happen with regard to predation? Well, the blue individual is very, very, very likely to survive and reproduce. So this individual is very likely to survive and reproduce. And it's going to pass on to at least some of its offspring, that B allele, right? So some of them, they're probably going to be homozygous Rs, but some of them are going to get that B, okay? Whereas the red individuals are going to, many of them are going to fall to predation. They're much more likely to die due to predation because they're so visible relative to the blue individual. So they're more likely to die. So they are less likely to produce reds, produce offspring. So then in our first generation, the first dots that I drew and had that one blue individual, we had one blue allele in the entire population, just one. One blue allele. That's it. But then in our next generation, now we have 3 blue alleles. So we've increased our P. We've increased our frequency of the blue allele. And we could go through and add up the numbers if we wanted to and figure out delta P. So the frequency of the first generation of the blue allele would be all of the alleles, the blue allele divided by all of the alleles would be its frequency. And now we can go to the next generation, and that would be the frequency of the blue alleles divided by all of the alleles. And we'll see that the frequency increases in the second generation. And then if we took this further, imagine these individuals who carry the blue alleles. So now we have 3 of them in the population. Okay, and they're going to make more offspring than do the red individuals. So then our P for that generation has increased, and then our delta P is going to increase. So in this scenario where we have a very, very strong, very big relative fitness difference. So that blue allele confers big relative fitness difference. We also have a very big S and strong selection. And so we would expect the frequency of this blue allele to increase over very few generations, to go up very quickly over very few generations. And if we start out at a very low frequency, whenever that allele reaches 1, that would mean that it's the only allele now. So the red allele is gone. We call that being selected out, and we call this fixation. So when the frequency of an allele goes all the way to 1, meaning it's the only allele in the population, we call it fixed. That's fixation. Okay. So this is how this allele spread works, kind of using a very simple, silly example. So hopefully that kind of helps solidify those concepts that we were thinking about. And remember that a very large difference in relative fitness is going to correspond to a very large S. So we have strong selection. Okay? But also another thing that you saw in your simulation was that if we include the influence of drift and we start with a really, really low initial frequency, that we can lose that allele no matter how beneficial it is. So if we imagine that this blue individual here, there came along, whoops, that's not what I wanted to do. Who knows what? A tornado. There was a tornado. And this blue individual died in the tornado. Not because they were blue, not because of any characteristic about them, but because of random chance that none of this is going to happen. No matter how beneficial that allele is, it's not going to spread in the population. And so that's what you're seeing with those wiggly red lines in the simulation. Okay, so moving on to thinking about how do we influence relative fitness? Not us, but how does relative fitness change? And one of the ways is through mutation, the introduction of mutation, but another way is through environmental change. So this is a really, really familiar example, the peppered moth. And you know from your previous studies, from previous learning, I think, that what happened in the pepper moth is called industrial melanization. So when factories were built and they were belching smog out into the environment, they covered the trees and they made the trees darker. And so this moth became more visible to predators than did the darker moth. But both of these morphs, both of these phenotypes were already present in the population. So there was already dark moths and there were already light moths. They were already there. Okay? But whenever the trees became dark, it became more advantageous to be dark because the environment changed. Okay, the alleles didn't change. The alleles were already there. Okay? The alleles that confer dark mothness and the alleles that confer light mothness were already present. The environment changed, and the change in the environment changed the relative fitness of these two alleles. Okay, so the alleles that confer dark now have higher relative fitness. So what do we see? We see the spread of that allele through the population, and now there are many, many more dark moths than there are light moths. Another example that we've already seen from previous discussions is pesticide resistance. Okay, so when pesticides were introduced, we changed the environment of these insects. So the variation that made them resistant to pesticides was already present. It was already there. But it was neither being selected for nor was it being selected against. It was just there. When humans changed the environment, we changed the relative fitness of the allele that confers pesticide resistance. So we changed the relative fitness. We changed selection. So selection on that allele became strong. And so then what do we see? We see that allele spread through the population because now it's adaptively advantageous. Okay? So those are examples where what was happening was selection was acting on what we call standing genetic variation. Okay? The genetic variation was already there. The different alleles that did the different things were already present. But they weren't necessarily, one wasn't necessarily more advantageous than the other, or the one that was previously advantageous now becomes disadvantageous due to a change in the environment. So like in the case of the peppered moths. So this is standing genetic variation. So that means that variation was already there. Okay? Now let's think back, let's pull in our previous learning about evolution. So we think about macroevolution and microevolution. So macroevolution is what produces that standing genetic variation. So change over long periods of time produces that standing genetic variation on which natural selection can act in the case of an environmental change. Okay. So we can get big changes in relative fitness without changing anything in the genome. We can get big changes in relative fitness based on changes in the environment. Another way, the one that we're probably more familiar with, is the introduction of a new mutation, the introduction of a new allele. And this happens through mutation. Okay, an example of this is that around 10,000 years ago, humans domesticated cattle. So around 10,000 years ago, I don't know if I can fit that, cattle domestication. And what this did was this introduced a new very rich food source, a new rich resource for nutrients, and that was milk. But humans, the enzyme that digests milk didn't persist into adulthood. So only babies had this. Because only babies were ingesting milk, right? Babies had this enzyme, this lactase enzyme to digest milk because they were nursing from their mothers. Well, about 4,000 years ago, in European ancestry, there was a mutation. Okay. And this mutation introduced lactase persistence. Okay, so around 4,000 years ago, in the European lineage, a mutation occurred that introduced lactase persistence. And so what this means is that individuals who carried that mutation could continue to digest milk into adulthood. If we think about 4,000 years ago, right now, if you can't digest lactate, if you can't digest milk, you're fine nutritionally if you live in the United States. 4,000 years ago, food was scarce. People starved to death often. And so this ability to digest this rich food resource swept through the population, because people who could do that lived, and they were healthy enough to reproduce, and so they produced more offspring, and then their offspring who carried this ability then produced more offspring. Another example is skin color. So if you look at this map, what we're looking at is indigenous skin colors. So the skin colors of the people who are indigenous to that area. And modern humans evolved, we know, in Africa. And I want to make sure I get the numbers right. And this area, Africa, is near the equator. There's very intense sunlight. And so this increased melanization of the skin was protective. Okay, this darker skin was protective against UV radiation. But about 60,000 years ago, and this is debated, so you may see other numbers, migration started to happen. Or I shouldn't say started to happen. There were earlier migrations, but this was like a major migration. Major migration up into northern climes, so into northern parts of the world. So away from near the equator, up into areas that these receive less sunlight, right? So these areas, so we got lots of sunlight here. And considerably less sunlight here. Okay. The major hypothesis about why humans evolved lighter skin color, why that was beneficial, why that swept through the population, is because of a condition called rickets. So this image right here is showing you an x-ray of a child with rickets. So rickets is because of a vitamin D deficiency. Vitamin D is necessary for our bodies to use calcium. Calcium bone growth, calcium muscle control, calcium nerve function. Calcium does all sorts of critically important things that you hopefully learned about in physiology. So vitamin D deficiency means our bodies can't use calcium. Well, how do we make vitamin D? Well, we make vitamin D from sunlight, primarily. Now we get it a lot in our food, but these ancient populations, it would have been sunlight. So the primary working hypothesis is that as populations went north, they started to develop rickets more and more. And rickets affects the bones. It can affect muscular contractions. It can have a neurodegenerative effect, so people's minds don't work correctly. And let's see, I want to make sure I get when this happened. Okay, so there was a mutation that occurred. I don't think I know when. So sometime after this migration, a mutation occurred in a gene called SLC24A5 mutation. Okay, and what that mutation did was it decreased skin pigmentation. Decreased skin pigmentation. So let's think about that. This mutation that drastically decreases the pigmentation of skin, and it spread very quickly through the population. It spread super quickly through the population. So why? Because we also know that individuals who have decreased skin pigmentation, add another, oh, that's not what I wanted to do. Okay, apparently, yeah, it's not going to let me do this. Hang on, just a second. Okay, so another thing that we know about lighter skin color is that there's an increased risk of skin cancer. So if you have lighter skin, you have increased risk for skin cancer. And as a matter of fact, darker skin has a sun protection factor increase of 10. So individuals who have darker skin SPF, that's what it actually means, sun protection factor. Their sun protection factor is 10 higher than lighter skin. So let's think about this mutation, this lighter skin mutation spread rapidly through the population. It has the markers, and we're going to talk about what those markers are in a minute, but it has the markers of super rapid selection, rapid spread through the population. So that means that it increased relative fitness. So you have lighter skin, you have this lighter skin mutation, so that means it had to have increased relative fitness. But we know that it also increases the probability of skin cancer, and darker skin has a much higher sun protection factor. So let's think about why. Why would this mutation spread even though it increases the likelihood of skin cancer? Think about when each of these things occur. So I forgot to tell you that rickets is a childhood illness. So people with this vitamin D deficiency who develop rickets, they develop it as children. And then let's think about when does an individual usually develop skin cancer? That's usually later in life. So when are these two evolutionary pressures occurring? The rickets is going to occur before this individual is able to reproduce. So if this individual is not able to get around, this individual has intellectual deficiencies, has muscle problems, maybe is physically different looking, they're going to be less likely able to reproduce and pass on their genes. Whereas the individuals who don't have rickets are going to be more likely to be able to pass on their genes. Then if an individual develops skin cancer and they develop that skin cancer later in life, so later in life, maybe at this point in human history is like 20, at that point, they've likely already reproduced. So they've already passed on their genes. And so this is sort of another thing when we're thinking about relative fitness. Remember, relative fitness is about whether or not you pass on your genes. So things that occur later in life that kill an individual aren't going to affect relative fitness. Okay. So how do we know, what does it look like when something spreads, a new mutation spreads very rapidly through the population versus when selection acts on standing genetic variation? Okay, we're at 23 minutes, so we're going to come back to that.